1308 web series, Chapter 5, The Working Cell. Now, as we noticed in Chapter 4, where we had to learn the parts of the cell and what those parts do, this chapter was going to take the next step and show you how things happen in the cell. So we're going to start off with Chapter 5, this first slide. Now, the reason why this slide is there is to show you that there are all kinds of things in the environment that actually can stop chemicals from working properly in our body. So this one right here shows you that nerve gas and insecticides, pesticides, they actually work in the body by crippling certain vital enzymes. So if there are certain chemicals that are supposed to do their job and poisons out there stop the cell from doing it, then we have issues. So the first thing we're going to see here is this slide, and this one's important. It says harnessing the cellular structures. That's what we did in Chapter 4, where we learned the parts of the cell and what they do. Now, here are the things that we have learned. You can think of a cell as a machine. So we have different parts of a machine, and they all have different functions. So some part of the machine may be for movement, some part of the machine may be for energy processing, and some are for production of various products or making new things. And that's what we saw when we made our flashcards for the animal cell, for example. We learned that the Golgi apparatus or the Golgi complex did the sorting and the packaging and the shipping kind of thing. The ribosomes, well, they are the ones that made the proteins, that kind of thing. So in Chapter 4, we learned what the parts of the cell do, and now we're going to see how we make use of that. So yes, in order for us to move with Chapter 5, you do have to have your flashcards made from Chapter 4, and that way you know what's going on. So in this example here, we see that cells control the chemical environment inside. Yes, they control the energy. They control how enzymes work. And remember, enzymes are often protein-based. So we learned another theme earlier, and as if you noticed, there's a lot of slides that repeat in terms of themes. And one of our themes we've learned was structure affects function. So if you mess up the enzymes that are supposed to do a certain thing, then those enzymes don't work anymore. Then the other one was cell membrane or plasma membrane. We also learned that the cell membrane is very selective. It allows certain things to go through and does not allow other things to go through. So for water, for example, in order for water to get into the cell, we have to have special channels. Well, that's exactly what this is like too. Okay. All right, next slide. This is pretty much the cell structures we saw in Chapter 4. And now here's where we're going to start seeing how the cell actually works. So the first word we have here is energy. And energy is defined as the capacity to cause change. A lot of people call this work. So if you were taking a physics class, they would call it work. Here in biology, we're calling it energy. It's defined as the capacity to cause change. And notice even the author here uses perform work, because that's what the physics class would use, perform work. Okay, so energy is defined as the capacity to cause change or to do something. Then we have these two forms of energy. Very commonly asked question on an exam. Kinetic energy is energy of motion. So when you push a ball down a hill and the ball is rolling down the hill, it's got kinetic energy. When the ball is at the top of the hill, it has potential energy. And usually potential energy is based upon location or structure. So in this case, potential energy, in my example with the ball on the top of the hill, that's location. In biology, you have structure as a potential, like ATP. ATP is the energy source that the cell is going to use. Just like a battery is an energy source for our cell phones. So cell phones use battery as an energy source. Our cells use ATP as an energy source. So one way or the other, energy is converted from one form to another. And that's exactly what that third sentence says. Life depends upon countless similar conversions of energy from one form to another. So when we eat our food, we take chemical energy, we convert it into kinetic energy when our muscles are actually moving. Okay, next slide. Again, this is a slide that we usually have our themes. And now we have this picture. And this is pretty much the same example I gave. While the person's on the diving board, they have potential energy. As they're jumping off the diving board, it changes over to kinetic energy. And then when they climb back up again, as they're climbing back up, they're taking their kinetic energy and storing it back again as potential, because this is a location issue. So we're storing up our energy as potential energy for the next dive. Okay, and this is a law that people will be asked. Matter of fact, I believe this is one of my clinical questions. So if you by chance randomly get this question, you'll know it. A physical principle known as conservation of energy, where energy is not created nor destroyed, but simply changes form from one form to another. Okay, next slide is heat. Now, heat is actually a wasted energy. So write that down if you haven't. So heat is a wasted energy. So it's a form of kinetic energy in the form of heat. So when you rub your hands together, you actually produce heat, but that's a wasted energy. When you're outside in the cold weather, to keep you warm and to keep you hot on a cold day, the fuel of the food you ate and you moving around is producing the heat, and that's the kinetic energy keeping you warm. So that's the first two lines. So all energy conversions generate some form of heat. So all living animals have some form of heat. Even a plant has some form of heat. By it's doing its metabolic stuff, it's producing heat. Then we have a word called entropy. And entropy is a state of disorder. And it turns out that in order for us to convert one form of energy to another, we have something called entropy. We have to have a state of disorder or randomness. It is much easier for your room to get messier over time than it is for it to stay neat. Because as you do something, the act of doing something is going to create some randomness, some disorder. If you did absolutely nothing, so if you never participated at all on Eagle Online, and I see the activity counter not moving at all because you didn't visit it, and then of course the entropy would be low because you didn't do anything. But as you are actively doing things on Eagle Online, actively moving around, actively doing and participating, you're going to see that energy has to be used, and that energy use is going to create entropy. And that's what that last line says. So every time energy is converted, entropy increases. Okay? So if you have a neat room and you never use your room, then it's going to stay neat, but again, no energy was used either. But as you use your room, it's going to create entropy. Okay, next slide. Okay, this is chemical energy. We've already talked about it. So gasoline, food, other fuels, they're all forms of potential energy. So the food you eat is potential energy. Gasoline in a car, potential energy. Okay, right? So other kinds of fuels like hydrogen fuel. There are cars being built with hydrogen fuel. That's a form of energy. We have EV cars. That's a form of electrical energy being converted to mechanical energy eventually. So yes, we have some form like that. And then here, the line here says living cells and automobile engines are the same idea. They take chemical energy stored as their fuel for work. The next slide is very important. This is the one that I use a lot in class. So here we have a car example and we have a cell example. In the car example, we have octane, like the gasoline numbers you see at the gas station, the 88, the 90, the 92 on the gas station pumps, right? So on the gas station pumps, you see those numbers. That's octane, and that's gasoline with oxygen. So you're burning it, you're producing combustion, and that's going to produce carbon dioxide and water. And of course, the wasted energy is going to be in the form of heat. Well, we're no different. We take glucose from our food. then the cells use it in the form of ATP. Remember, we have to convert it. Just like we go to another country, we convert our US dollars to whatever euros for Europe, for example. So we have to convert our currency. Well, that's what we're doing here. We're converting glucose currency to ATP currency for the cell. And again, we're going to have in the cell wasted energy in the form of heat, and we have the same products, carbon dioxide and water. So anything that's a combustion, and we are a form of combustion, we are like a furnace. And the more you fuel it, the more energy you have for fueling it and burning it. If you don't have much food energy, you're not going to be as active, and you're going to be sluggish. Sluggish both outside as well as inside your body. So carbon dioxide and water, carbon dioxide and water. Please make a note of this slide. You do need to know the products here. Carbon dioxide, water, carbon dioxide and water. Okay, next slide. Okay, this slide here just lets you know that how efficient we are. While humans only convert about 34%. The rest of it goes in the form of heat. So we're not as good as some cars are. We're not as good as some other mechanical things. We are not great. Most of our energy goes in the form of heat. And that's why we stay at 98.6 degrees. Because we are warm-blooded animals, and we're warm-blooded because of the fuel that we eat to stay what? Warm. Okay, next slide. All right, these are numbers just to be aware of. Calories, what we measure food calories in. So when you're reading your nutrient ingredients on a food package and they say we have so many calories, well, those calories are actually kilocalories that you see on there. So when something is 200 calories on a wrapper, like some of your nice satin cream pies that you find as desserts, and it's like 320 calories. Well, those 320 calories are really kilocalories. So you should actually be adding 1,000-fold to that. So it's not just 320 calories, it's really 320,000 calories, really, because food calories are always in kilocalories. Okay, all right, next slide. Okay, here's an example, just to show you. Here's that dessert example I gave you a moment ago, like the satin cream pie that Marie Callender's has. And so that's around 320 calories, but it's really calculated as kilocalories. So it's 1,000-fold more when we really look at it. Okay, next slide. This is now ATP. I've been harping about ATP for some time now. So this right here is to let you know that this is the cell's energy form. And ATP is called adenosine triphosphate. And ATP is used in many ways within the cell. As you can see, it says it acts like an energy shuttle. So it's actually an energy storage form for the cell. All right, next slide. So energy storage form for the cell. Okay, next slide. Here's how ATP power works. Here is the energy in ATP. It's stored in these phosphate groups. So ATP is the energy, but it's stored in the phosphate groups. So as you release energy, you actually pop off a phosphate group. Okay, let me say that again. So as you release energy, so as you make energy, you're really releasing a phosphate group. Now this phosphate group that has popped off, that is going to attach to something. And we'll see that as we go along. So when you take off a phosphate group, that phosphate group is going to attach to something. And whatever it attaches to, it provides energy to it. So let's take an example of a door. Let's say a door is unique. So you need to have ATP to open up this door. So what you do is you pop off a phosphate group. And that phosphate group will attach to the door. When it attaches to the door will change shape. By changing shape or form or structure, we've learned that it changes function. And then that door will change shape and then it will be opened. And therefore, it's now opened. And then things can go in or come out or whatever's supposed to happen. Then when you want to close the door, you remove the phosphate group. And when you remove the phosphate group, the door changes back in shape and structure to the original shape and structure, so it now is closed. That's how we do things in the body. So earlier I mentioned channels that we have in our phospholipid bilayer of the cell membrane. In order for that channel to open, we actually attach a phosphate group. We will see a picture of that later. So you're opening up a channel by attaching a phosphate group. So when cells need to open up a channel, they actually pop off a phosphate group from ATP. That phosphate group attaches to the channel. The channel changes shape and form. It opens up like a channel. Things can go through. And then when you want to close it, you just remove the phosphate group and the door and the channel will close back up again. That's how things work in the body. Okay, next slide. Now, this slide says exactly what I said in words a moment ago as a description. What does that energy do? It changes the shape of something. It changes the action of something. It changes the structure and therefore the function. Now, the next picture says it best, and that's this one. We're going to go through all three of these to prove a point. Here's the first one at the top. This is how muscles work. So this is how muscles work. And notice I want to crank this arm. So I want to crank this arm. But the only way I can crank that arm, so if you look carefully, you'll see that this arm is actually bent here. It's cranked. And the way I do that is adding a phosphate group. When I pop off that last phosphate group from the ATP, when I pop off that last phosphate group and I attach it to this little arm, the arm will then bend, change shape, and then it's going to move the protein of the muscle, and that's how our muscles actually contract. Okay, so things happen because we are changing the shape of something. So in this case, it's mechanical work. And we're doing that by adding a phosphate group that came off of the ATP. And then when I want to make it go back to the original shape where it's not bent anymore, I remove the phosphate group. Same is true for the next picture. Here we want to let these marbles, these purple marbles, go through this channel. So we want it to go through the channel. And as you can see here, we added a phosphate group. And by adding a phosphate group to this, it opened up the channel, allowed the marbles to go through, and that marble can be anything, allowed it to go through. And then when I want to close it, notice what I'm doing. I'm going to remove the phosphate group, and this channel will now close. Okay? So that's how that second example works. That's how that channel story works. And then down here, we have a chemical reaction. We want to join X and Y together down here. Well, in order to join X and Y, somehow this has to change shape, where X and Y can now be a lock and key kind of thing, where it joins together, okay, like a lock and key. And the only way to do that is to add a phosphate group. It changes the shape of X so that X can now join with Y. And then once it's joined, we remove the phosphate group. And that's why this slide is so helpful, because it explains that the phosphate group is where the energy really is in ATP. And when we attach the phosphate group, it makes a structural change. It allows that item to do something. And then when you want it, once it's done and you want to remove it, then you remove the phosphate group and then the product is there. Okay? So this picture is very important to study. Next slide. Okay, and notice we have lots and lots and lots of ATP. Matter of fact, one glucose, and this is something I'm giving you as a heads up as we go throughout the other chapters. one glucose makes about, and I'm using the word about because there are variations as we'll see later, but one glucose makes about 36 ATP. So one glucose makes about 36 ATP. So every time we use a glucose in a cell, when the cell uses the glucose, it makes about 36 ATP. Next slide. All right, so here's our story again. We start off with glucose. Glucose produces ATP. We use the phosphate group, and that produces energy, and then we eventually recycle the whole thing. So more energy from glucose goes in to make ATP. ATP releases a phosphate group. That makes energy for us to do something with, and then we again put these two together all over again. So it takes glucose to make ATP. Next slide. Now we come to enzymes. Now enzymes are like catalysts. So if you were taking a chemistry class, this would be called a catalyst. Here we call it an enzyme. And what enzymes do is they speed up the reaction. They do not get used up though. So they're not broken down, they're not used up. All enzymes do is they speed up the reaction. Now notice it says here, enzymes are proteins. That is a very important side note that you should have noticed as we went along. Okay, so as we went along, you should have noticed that enzymes are made out of proteins. So as you can see, as I'm going through these lecture videos, I'm giving you themes along the way. So enzymes are made out of proteins. And yes, they speed up the reaction. Now, why are enzymes so important? Because without enzymes, a chemical reaction may take years to happen. And with the enzyme, it might only take seconds to happen. Now, I've firsthand experienced this as a college student where we had messed up on an experiment. Now, the experiment was messed up because the instructions were wrong for the entire chemistry department. So every class that was doing this experiment, every class for every instructor, the experiment was messed up. So then the professors went back to the drawing board and they figured out what was wrong. And it was critical because the end product of that experiment was supposed to be used throughout the semester. So if we didn't get this right, then the other experiments in the semester wouldn't work either. So the professors went back to the drawing board and figured out that the enzyme was incorrect. That's what I'm getting at here. A specific enzyme is needed. Not just any enzyme will do. So if you remember in a previous chapter, we had the lactose intolerance thing about milk sugar. It took special lactase to break down lactose. Well, in the same way here, it takes a special, specific enzyme to do the job. And if you have the wrong enzyme, the chemical reaction may even take years to happen instead of seconds. So when the college chemistry department finally figured out the mistake and we used the correct enzyme, that reaction only took seconds instead of years, as they would have said it would have taken. Okay, so that's how enzymes are. They're very, very specific, and their job is to speed up the chemical reaction. Okay, moving on. Okay, now there's a definition here called activation energy. Now, activation energy is the energy it takes to go from reactants to products. That's one way to say it. So activation energy is the energy it takes to go from reactants to products. Now, another way that I come up with it is this. Activation energy is the energy it takes for the reaction to even happen. Because it's a starting nudge, a little push that it takes to make it happen. So for example, my checklist that I provide at the end of the week. So you have noticed that the first week of our semester, I provided an update because I saw very little activity on Eagle Online. Okay? Because by the end of the week, I should have seen hours, not minutes, for Eagle Online activity because you need to get going in order for the semester to roll correctly for you. So that was a little nudge by giving checklists. I provide checklists at the end of the week so that way you know what you need to finish by then. That's what this is. Activation energy is the energy it takes to go from reactants to products. It's that little nudge that it takes for a chemical reaction to happen. Now, sometimes the activation energy, a great deal of activation energy is needed. Sometimes very little activation energy is needed to make it happen. Okay. All right. And the next slide will prove that point. So in this picture, we see picture A. Picture A shows you this long, high wall. And it says reactants turning into products. But notice because of this high wall, which is the barrier, which is that activation energy, it's going to take a lot of activation energy for reactants to turn into products. It's less likely to happen because there is no enzyme. However, if there is an enzyme present, look what happens to the wall now. The wall is lower. So by adding an enzyme, you lower the activation energy. That's the type of test question you would see. What does an enzyme do? It lowers the activation energy. So enzymes make it more likely that the reactants will turn into products. That's what it really does. It just provides that extra nudge, that extra push to allow reactants to easily turn into products. Okay. So enzymes lower the activation energy, and it makes it more likely that reactants will turn into products. At the end of the semester, if you actually read the day one announcement very, very carefully, at the very, very bottom of the day one announcement, there is an offer for test number 3. And that offer is 5 points added if the class reaches 80% compliance. That means as a class, 80% plus people have to complete the EGLS 3 at the end of the semester. And that's at the very bottom of the day one announcement. Now, when I do that, when I make that kind of offer, I see the compliance easily get past 80%, usually like 95, 98% compliance from the class because they want that 5 points added to test number 3. When I don't make such an offer, I don't get that 90%, 95% compliance. I might get 30%, 40% compliance. So that offer on the EGLS 3 survey, that offer is acting like an activation energy enzyme, where the offer makes it more likely that the students will complete the survey because of the offer. So that's the example I give there. That is the activation energy for students to complete a survey. Okay, so that's exactly how this works. It lowers the activation energy. Okay, next slide. Don't care. Don't care. All right. Now, this is important because this has to go back to what enzymes are. Remember, we said this several times. Enzymes are made out of proteins. If enzymes are made out of proteins, that means they must be coded for in the gene. And the gene, of course, is what again? Which I've said several times as a definition, which I'll be repeating all the time. Genes are a sequence of DNA that codes for a specific protein. So if you have a mutated gene, and in this picture here, you see that there is a blue line where that's the gene for the enzyme. But then all of a sudden, there's something wrong with this mutation here. There's something wrong with all these oranges. That means that somehow these enzymes are now changed. The protein structure of this enzyme is changed. And now you should see a theme here. If I've changed the structure of the enzyme, it means that it might not work anymore. Okay? And that's why it's so important that if you mess up the gene for an enzyme, then that enzyme won't work. Now this goes back to that lactose milk intolerance story. Remember I said Asians tend to have this gene for lactase either poorly developed or not working. So if the enzyme doesn't work as lactase, then it doesn't break down lactose. And then you're going to have the problems with lactose intolerance. So that's why there's a defective gene for that defective lactase enzyme, and that's why it's not working. Okay? So those people have a genetic problem, and that's why the enzyme doesn't work. Okay, next slide. Okay, now we have this same story again. As you can see, a theme building up, structure and function. Now, an enzyme has to act on something, and what the enzyme acts on is called a substrate. So the enzyme acts on a substrate. So an enzyme will act on a substrate. Now, this slide is nice, but there's a better one with a picture coming up. Okay, again, I don't care for this either, but I do care for this picture. So this picture is basically the last few slides summarized for you. Here we have an enzyme, and we are using our classic one again, lactase. So here's our lactase enzyme. And then here's our lactose, which is the substrate. So whatever the enzyme acts on is called a substrate. And in this example, it's lactose. So whatever the enzyme acts on is called a substrate. And notice here, what's it doing? It is taking these two enzyme pieces here and breaking them apart. Now, if you are keeping up with your notes, you should know something here. Remember, what shape are these? These are glucose and galactose. And glucose is a monosaccharide from a previous chapter. This is glucose monosaccharide, a monomer. And how do I put pieces together? I put pieces together with dehydration. Now, since we're not putting it together in this picture, we're splitting it apart. How did I say in a previous chapter slide, previous chapter lecture video, how do we tear it apart? How do we break up the pieces? We do it by hydrolysis, by adding water. So if you look carefully, and I know you probably can't see this because these things at the bottom of the slide are really sort of instructors' notes for instructors to use when they are presenting a PowerPoint, like in this example that I'm doing here. It says in step number 3 down here, I know you can't see it, that's why I'm reading it for you. It says, with the addition of water, we have hydrolysis, and we are going to break it apart. And that's what we saw in a previous chapter. So hydrolysis adds water and breaks the pieces apart. And that's what this enzyme is actually doing. So we broke these two pieces apart by hydrolysis. So now you see how we're putting chapters together. Okay, next slide. And you can see here the author is using the word catalyst because enzymes are a catalyst. Okay, next slide. Repeat theme. All right, now we have something called enzyme inhibitors. This is where we start having issues with diseases. So enzyme inhibitors. Okay, so this is where diseases come into play. Okay, all right, next slide. So again, that's the title, enzyme inhibitors. None of this makes sense until we see a picture. So I'm a picture person, so we'll see it this way. So there's several examples. The first example of enzyme inhibitor is this. Here we see a normal situation where an enzyme sticks to the substrate. Normal situation. Enzyme sticking to a substrate. No problem. Then we have an enzyme blocking a substrate. So notice here, here's an enzyme. But we see something called an inhibitor. It is actually blocking the substrate. Think of this like what you did when you were a little kid playing musical chairs. If you remember how musical chairs is played, where the music stops. So when the music stops, people that are standing up try to sit down. And anyone still left standing because there's one less chair, anyone still left standing is the loser. Well, that's exactly what this is like. In this example, the inhibitor, some other chemical, it could be a poison even, and that's what it is usually in the real world. some kind of poison sat there first, and therefore the substrate wasn't allowed to do its job. And that's how some poisons work, like the pesticides, the insecticides, that sarin gas or VX gas that was mentioned at the start of this slide, start of this chapter. So there are poisons out there that actually block. And so they'll block the spot, and the real substrate, the real thing we're supposed to do, doesn't work. Okay, so that's one way. Another way is where the enzyme changes shape. So you have some kind of poison that doesn't block, so it's not a blocking thing like letter B over here, but it actually changes the shape of the enzyme. So here's an inhibitor that comes in, changes the shape of the enzyme, so now the opening is not the same. The active site is no longer the same. See how much smaller it is now. And since it's so much smaller, where we changed the enzyme this time, the substrate can't do its job, or the enzyme can't do its job. So see the difference between the two. And here we have a third party sitting there, and therefore the substrate can't do its job. And over here, all we did is change the shape of the enzyme. So two different ways, and all key poisons work this way. They either block or they change the shape of the enzyme so the substrate can't work. Next slide. Review a theme again. All right, I've already mentioned this. I'm going to show it to you in a picture form. I already talked about the cell membrane, and I already talked about the proteins. So let me go ahead and go to that slide, which is the next one, and that's this one right here. So here we see our cell membrane, and we see all these things that are embedded in it. And if you recall earlier, I mentioned that these membrane proteins play several roles. One of them is a channel. Now, some authors call it a transport. It's the same thing. So a channel or transporter, that's one of the functions of a membrane protein. So one function is a channel. The second function is cell ID. And I mentioned that only identical twins would have a similar cell ID as a marker on your cell. Your siblings that are fraternal twin siblings would not have the cell ID being the same. So this is where organ transplantation comes into play, where they have to match as close as possible to tissue so that there isn't a tissue rejection. And that's why people who do get other people's organs, they have to take immunosuppressive drugs to prevent tissue rejection. If you have a very close cell ID match, you're going to have less tissue rejection. That's why they check out to see who's the closest match. So cell ID and transporter. And the last one I gave you before that I'm now repeating again today is receptor. So they're not showing you a picture of that, but this enzyme example could serve as a receptor. Some people call cell signaling here as a receptor as well. So a receptor. And at that time, I mentioned receptors are how chemicals, drugs, and even hormones, that's how they know where to go to. A receptor on a cell. And I mentioned the insulin example at that time, where insulin knows where to go to because there's a receptor for it. So receptor is one function, cell ID is one function, and channel and transporter is one function. Okay, next slide. Okay, now we go into how things move across. So how things move across. This is where it's best to draw a line first, because this is a topic that usually chapters end with as a different thing. So this is a separate category. Now the first thing we're going to write down on our paper is passive. So I'm going to give you definitions here to make sense of this, because the slides always don't do a good job here. So passive transport. Passive means it does not require ATP. So passive does not require ATP. The notes I'm giving right now are critical for understanding the upcoming pictures. So passive means that it does not require ATP. The second thing, passive means it's going down the concentration gradient. Now, what does that mean? Down the concentration gradient. It's going from high concentration to low concentration. So from high concentration to low concentration. That's down the concentration gradient. So we have two things on our paper so far. Passive does not use ATP, and it goes from high to low, which is down the concentration gradient. Now, the best example, which you see on this slide, is diffusion. So that's your first example for passive diffusion. Now, the example that you'll see either for me or if you're taking the lab version or something like that, what they do is they take a glass of water and they put a food coloring in there. So when you put a drop of food coloring in a glass of water, you see that the food coloring slowly permeates into the glass of water, and eventually the entire glass of water is that one color, whatever that color is. But it takes a while. But it's going from high concentration to low concentration as it permeates through the glass of water. So that's diffusion. Okay, next slide. Now, here's an example of diffusion, and we're going to pick the top example first. So here we have marbles on the left-hand side, and then we see some kind of a membrane, and then we see very little or nothing on the opposite side. And that means that the marbles are going through this membrane from a high concentration on the left to the low concentration on the right, just like the food coloring dye that goes in the glass of water. So it's going from high concentration to low concentration. And eventually, as you can see on the far right, it reaches equilibrium, where we have the left side equaling the right side in terms of the coloring. And that's exactly what's going to happen when it comes to the glass of water with the food coloring. Eventually, the food coloring will equally fill out all spaces of that glass of water. So it'll be in equilibrium. Then on the bottom picture, we see now two different colors. But again, they're going to move in their respective high to low. So you see the purple ones or the blue ones going from left to right because it's moving from high to low in that direction. And then the red ones are going from right to left because the concentration for the red is from the right to left, which is high to low again. And eventually the red and the blue will even out in reaching equilibrium. Okay, so this picture is worth studying as well. Next slide. Okay, now we have another kind of diffusion, but it's called facilitated diffusion. Please make a note that it's still under passive. So it's still under passive. That means it still goes from high to low. So facilitated diffusion is still under passive. It goes from high to low. Now the only difference here is that it requires a carrier. So somewhere on that cell membrane is a carrier, a transporter like we saw in a previous picture. Now, the best example I can give here is sandbags. Let's say you have a wall and you want to put sandbags from one side of the wall to the other side of the wall. And what you're doing is you are serving as a carrier or transporter that's built into that wall. And you are now doing what? Moving or tossing the sandbags from one side of the wall over to the other side of the wall. So yes, you are a carrier, you are a transporter, but yes, you're moving it from one side to the other as passive. No energy is required, but you're a transporter or carrier that's doing that. And you're moving it from high concentration on one side where the more sandbags are and tossing it over to the other side where there's less sandbags. And I'm saying that to you this way because usually there's a picture at the end of the chapter, not here yet. Next slide. Okay, the next slide is osmosis. So we're still on passive. So we're still adding another bullet to passive. So we had diffusion, so far under passive. Then we had facilitated diffusion under passive. And now we have osmosis under passive. Now, osmosis is the movement of water. Now, I'm going to give you a definition that I'll repeat a couple times and then we'll move on and see a picture for it. So, osmosis is the movement of water from high concentration to low concentration within a selective permeable membrane. So, it's moving from high concentration to low concentration within a selective permeable membrane. Okay, so high concentration to low concentration within a selective permeable membrane. So because of that selective permeable membrane, water is going to move from high to low. The best example is this picture right here for osmosis. Now let's look at what this picture is telling us. At the start, the water levels are the same. So at the start, the water levels on both sides of the tube are the same. But then we have a membrane here in yellow, a membrane. The membrane is very selective. It only allows water to move, not the solids. In this case, not the green sugar. So not the green sugar. The green sugar cannot move. Only water can move. And if you look carefully, we see less sugar. And here's what that means. It's a comparison. So if I have less sugar, by default, I have more water. And if you look at the left-hand side of this picture, I do have what? Less sugar and more water. More water in comparison to what? The other side. Because the right-hand side has more sugar and less water. So because of that, the more water side is going to move the water to where there is less water. And that is what osmosis is. In our body, we move water by artificially creating a water gradient from high water concentration to low water. And we typically do this by moving some kind of molecule. In this picture here in the PowerPoint slide, it's sugar. In reality, in our bodies, it's usually sodium. We actually move sodium in one direction, and when sodium moves in one direction, it causes water to go with it. One of the rules I've learned as an example is potato chips. And this is technically not the same thing, but it does help as an example. When you eat a lot of salty potato chips, don't you feel like you have to follow it up with water? Well, that's exactly how it works in the body as well. Wherever salt goes, water follows. And that's how we actually absorb water. If it didn't work in our gut that way, if we did not reabsorb salt in our gut, then water would not be reabsorbed and we would have constant diarrhea. So the reason why we have diarrhea issues is somehow salt is not being reabsorbed properly. So once we fix the problem where salt is being reabsorbed, water will follow and the problem will correct itself. So in this picture here, we see water going across because there's more water on the left, less water on the right. And that's why we see the water levels reaching a higher level on the right because the water went from left to right. Okay, next slide. Okay, now we have this slide. This slide deals with hypertonic, hypotonic, isotonic. I do not like this slide. It's best explained by a picture, and that is this picture. So let's look at a normal animal cell first, and then that will help with each of these. So in a normal cell, you see water going in at the same rate that water is going out. So we're looking at the animal cell only first. That makes better sense. So we'll start with the animal. And notice that's called isotonic, where the water is going in at the same rate that water is going out. In essence, water is really not doing anything because it's going in at the same rate that it's going out. And that's called isotonic. Now, where do you see this in the hospital setting? You see this with the IV bag. When they give you an IV, the water that's in the IV bag is in an isotonic situation with your blood. And remember, most of your blood is water. So the IV situation, the IV solution is in equal isotonic with your blood. Now, what if we have a situation where it's hypotonic, which is letter B here? Again, we're only looking at the animal cell to learn it first. Hypotonic means that water is going into the cell. So somehow water is going into the cell. Now, if this happens, eventually the cell is going to swell up and it's going to pop. It's going to burst. Now, that's called hypotonic. And how are you going to remember that for the test? Hypo equals hippo. And you know a hippopotamus is big and fat, right? So that's how you're going to remember it. Hypotonic is hippo. So hippo, hypo. That means the water is going into the cell and it's going to swell up. So if you have this situation happen in your body, you're going to find that the cells are swelling up and your body will actually bloat. Your face will swell up. So if they give you an IV and things aren't working properly in your body, and they're still giving you IV and they keep giving you IV, your body is going to swell. It's going to bloat if you're not able to maintain the water balance correctly. So hypotonic goes with the swelling, and eventually the swelling will cause the cell to pop. And that's what this picture here. Lysed. The water goes in and eventually swells it up. Then the next one is hypertonic. Hypertonic, where the water's coming out of the cell. And here the cell is going to shrivel up. Now this happens when your body is in a state of dehydration. So when you're dehydrated, that's what happens to your cells. They shrivel up. So this is now hypertonic, going with water coming out of the cell, causing dehydration. Water is going to come out and the cell is going to shrivel up. Make sure you understand the top part for the animal cell first, because that's the best way to learn this slide. Then, once you have the top understood, then you can see the exception that goes for the plant. The plant is unique. Remember, plant cells have a cell wall, so they're going to act differently. So in a plant, the plant is actually going to shrivel up. So if you ever see the leaves of your plant shriveling up, that's because it has too much water going in. So when too much water goes in, you're going to see the plant leaves go limp. So you're watering too much. So when your plant leaves are limp, you know it's watered too much. It's flaccid. Too much water is going in. Then, and again, that's isotonic for the plant. Then we have turgid. Turgid is when water is going in, but water's not coming out. Okay, so water is going in, and that's considered actually normal for the plant. So plants like to be in a state of hypotonic. That's why I said you need to learn the animal cell first, and then you go to the plant for the exception. So hypotonic is normal for the plant. Then we have, similar to us, we have shriveled again for hypertonic, when the plant is now having a problem with too much water leaving. When too much water leaves the plant, then that's going to shrivel up as well. So we see that story there. Okay? So make sure you're understanding this picture. Sometimes flashcards come in handy here. Okay, don't care for that one. Remember, let me back up. Remember, the plants have cell walls. Okay, so plants have cell walls. And again, the author says it nicely here. Plants like to be healthiest when they're in a hypotonic. And that's what you saw in this picture here. The normal is hypotonic. Okay, next slide. Okay, this is the one I had up just a moment ago. And now we go to the picture. So here is a turgid, normal, and here's where it's wilted, which is bad. Okay, next slide. Okay, we're now closer to the end of the chapter, and we see just the same stuff repeated, but this time we're doing the last part, which is now active transport. Active transport requires ATP. So active transport requires ATP. Okay, so here you can't do without energy. Active transport requires ATP. Now notice the paragraph says it goes against the concentration gradient. So this time we're going from low to high. We're going against the gradient. It's going to take energy to go against the gradient. The best example I give here is, let's say someone is trying to get into the movie theater when everyone else is rushing out of the movie theater. So if that one lone soul is trying to get into the movie theater when everybody is rushing out of the movie theater, it's going to get a little tough for him to get into the theater, right? Because everyone's rushing out and he's trying to push his way in. So that's an example of active transport requiring ATP, going against the concentration gradient, and that's from low to high. And here's a picture for that. So let's say we want to move something against the gradient. Notice there are more marbles inside and only one marble outside. So in this case, we're going from low to high. And in order to go against the gradient, we're going to need ATP. Now, if you remember from our previous information, ATP has the energy stored in the phosphate group. Not shown in this picture, but it's implied. So that ATP has that phosphate group. We take off the phosphate group. We attach it to this purple, which is either a receptor or a channel or a transporter. It changes shape, as you can see here, and it allows something to come in. And then we remove the phosphate group, and it allows it to close up again. So we're going against the concentration gradient, and it does require ATP. And that phosphate group from ATP is going to cause a structural change, allow the item to come in, and then we remove the phosphate group, and it goes back to its original shape. Okay, then we have these last two terms here. We have exocytosis, that is when we're spitting things out of the cell. So exocytosis spits things out of the cell. Then we have endocytosis, which is when we take things into the cell. So they're just opposites. Exocytosis removes things from the cell. Endocytosis brings things into the cell. One of them is called phagocytosis, and that's what your white blood cells do. So white blood cells do phagocytosis. And what that is when your white blood cells go around looking for germs. That's what white blood cells do. They go looking for germs. And once they find the germ, they actually eat it up. And that process of eating up the germ is called phagocytosis. It's a subset, an example of endocytosis. Okay? So let me repeat, then we'll finish up. So exocytosis is removing things from the cell. Endocytosis is taking things in. And one example or version of it is phagocytosis, done by white blood cells, where they actually take in the bacteria to destroy it. Now there's another version that's going to be mentioned on one of the slides, and that's called pinocytosis. Pinocytosis, it's the same thing, except instead of taking solids, it takes liquids. So pinocytosis is taking in liquids, and that's a subset. So phagocytosis takes in solids. That's what white blood cells do to bacteria, and pinocytosis takes in liquids. Something like a food vacuole would do. Okay, here's an example of exocytosis, where we're spitting these purple marbles out of the cell. So these purple marbles are being released out of the cell. That's exocytosis. And then we have endocytosis, where we're taking all of these green marbles into the cell. Okay? And that's how cells take in things too. Okay, next slide. I don't care for this. We've already mentioned it. I've already talked about phospholipids. I don't care. Okay, I don't care for this either. And that happens to be the end of chapter 5. It's the end of chapter 5. Please note that you now have chapters 1 through 5 that make up test number 1. You can find that information in the day 1 announcement, letting you know what chapters are found on the first test. And that will include chapters 1 through 5 at the moment.